This is Nora Berry from Process Development Forum, along with Amy Ritter from BioPharm International Magazine, and we're speaking with Parrish Gallagher, who has been the CEO and founder of Accelerex, which is now part of GE Healthcare. We're speaking today with Parrish about future trends and in fermentation and cell culture equipment. Parrish, thank you for taking the time to speak with us today.
Thank you very much.
Let's start right off by asking, what important new trends do you see emerging in approaches to monitoring and controlling fermentation and cell culture operations?
The most active trend right now is the application and use of single-use sensors for pH and dissolved oxygen to monitor and control the cell culture or fermentation operation.
And why do you think that has emerged, the single-use sensors?
It's emerged as a result of the development and advent of the single-use cell culture bioreactors and fermenters, which are now also in development.
What trends do you see emerging in equipment overall, in the fermentation and equipment overall?
Well, besides the single-use trend, we see an increasing sophistication in the control strategies for fed batch and perfusion culture operations.
Before we began the podcast, you had spoken a little bit about stainless steel and single-use bioreactors. Can you talk to us a little bit about how you see the balance between stainless steel and single-use bioreactors evolving over the next few years?
We expect that the penetration of single-use bioreactors will continue in the industry and will probably increase its rate of penetration over the coming years.
Okay. And do you attribute that to anything in specific?
Well, the penetration rate we think will increase because the technology is being hardened steadily by not only the vendors who supply these systems, but also by the customers in their work, testing out those systems and feeding that information back to the vendors.
How do you see growth in different geographical markets, for example, the BRIC markets?
Well, we see that growth just beginning now, and it's as a result of the improving economies in those territories. However, we see that the lower labor rates and lower cost of living result in a slower penetration of the single-use cell culture technologies.
And why is that?
Well, because the cost of cleaning stainless steel and sterilizing stainless steel systems is not as high as in developed countries due to the lower labor rates. So as a result, single-use technologies are more expensive, can be more expensive than stainless steel manufacturing systems.
Okay. So it's really tied to the economic culture of the region in many respects.
Right, which, as you know, is changing. So as those regions further develop and the cost of living increases, those territories are expected to turn into growth opportunities for single-use technologies. Okay.
Interesting. That's very interesting. Tell me, what is your view on the discussion of increasing titers in mammalian cell culture, especially for monoclonal antibodies?
Well, it's a hotly debated subject. There is a trend to drive to higher titers from the cell culture folks, but in point of fact, the downstream folks are struggling with the increase in titers, and especially in the single-use arena, single-use technologies are quite limited in large scale, so they are not suitable quite yet to handle these higher titers downstream.
So it's still mostly using the stainless steel equipment then?
Right. For larger scale, for very high titers, you have to use stainless steel downstream systems to accommodate the amount of product that's coming through the pipe.
Do you see that changing? Do you see an evolution in use of single-use bioreactors for increasing titers?
Absolutely. We see the development on the horizon, the development of high-capacity downstream purification systems that are single-use. These are in development stage now, and as they mature and enter into the marketplace, there will be a breakthrough in the productivity of downstream purification using those single-use technologies. And that's really what we need to keep up with the increasing titer and productivity of the upstream cell culture operations.
Okay, thank you. One final question. If you had a crystal ball, what would you say is the future of fermentation and cell culture equipment?
We see the scale of fermentation and cell culture equipment continuing to shrink, largely because of three trends. First, you already mentioned the increasing titers in the upstream space. Secondly, the potency of the new generation drugs that are in the clinic is increasing, which means the doses are lower, which means you don't have to produce as much, which means your facility is not as large. And the third trend is the trend of manufacturing in market for market. What we mean by that is a trend in the direction of smaller facilities distributed in many different small markets around the world, which obviously don't have as large a territory to supply and therefore do not have to be as large in their production capacity. So those three trends are combining and will continue to intersect and result in smaller and smaller fermentation and cell culture equipment going forward. All
right. Well, good enough. Thank you very much for your time today. We really appreciate it. And once again, we've been speaking with Parrish Gallagher from Accelerex, which is now part of GE Healthcare. Thank you, Parrish.
Thank you very much.